Hello, everybody. In this lecture, we're going to go over the basics of enzymes. So let's get right into it. By the end of this module, you should be able to describe the structural and functional features of enzymes and state why cells need them. Describe how enzymes lower activation energy. Give examples of coenzymes and their functions, and describe what ribozymes are and how they are different from typical enzymes. And finally, predict how environmental changes affect enzyme activity. All right. So here's just a little bit of an overview of enzymes. Enzymes are a type of protein catalyst. And what they do is they help the cell complete chemical reactions. And so in chemistry, catalysts are things that help a chemical reaction proceed, and they are not used up during that reaction or altered in any permanent way. And so these are going to act on specific substrates, right? So like DNA polymerase is going to act on DNA. RNA polymerase is going to act on RNA. And then they're going to generate specific products. These are going to be, when we've talked about throughout the semester, the cellular machines, these are those. Enzymes are the machines of the cell. They do the work. They often end in an ACE, right? And their name is often related to what it sounds like. So sucrase is an enzyme that breaks down sucrose. Since they're a catalyst, they increase reaction rate. They're not consumed or permanently changed. And so with that, let's talk about some kind of basic terminology we need to understand before we get into the details. And the first thing, and you may have seen something like this when you took chemistry, is activation energy. So our y-axis here is total energy, and our x-axis is the reaction pathway. We've got two states. We've got the reactant and the product, and then we got this intermediate called the transition state. So activation energy is the amount of energy that is required for a reaction to occur. And so, let me go back. For this reaction to occur, think of this ball needing to get up this hill. All right, so we need to get this ball pushed up to the top of the hill. When it gets to the top of the hill, it proceeds down, right? And so the amount of energy it takes to get to the top of the hill is the activation energy. Once the activation energy is reached, the reaction can proceed. Now, what enzymes do is they actually reduce that activation energy and increase the reaction rate. So instead of having to push the ball up this really steep hill, that hill becomes much less steep. And so you need less energy to get up here. And the cool thing is that these catalysts, these enzymes, aren't used up every time they go through this process. They can be reused over and over again. Now, reactions can be either endergonic or exergonic. And the cool thing about the enzymes is that they're going to actually increase the amount of energy gained or decrease the amount of energy lost. And so on the left-hand side, we have an exergonic reaction. And an exergonic reaction, what we're going to do is generate energy, right? We're going to break something down usually. Most of the time, these are going to be hydrolysis reactions. And it's going to produce energy. The energy that's being produced is the energy from the net production is from this state where the reactants start and down here where the products end. All right. Now, the gross gain, like the overall gain, is from the top up here all the way to the bottom. But there has to be an energy investment, right? So how do we get up to this transition state? So some amount of energy is invested, and then we get up to the top of the transition state, and we get all the energy that comes out of the bottom there. Oopsie. Now, when we have an enzyme present, we reduce the amount of energy to get up here. And so the net gain is greater. So if let's pretend that to get up to this point, that's 2 energies, And to get up to this to the enzyme point, that's 1 energy, right? And so what we're doing by adding the enzyme is reducing the total amount of energy invested, and we get the same amount of net energy gain. Okay. So on the other hand, we have inorganic reactions like this. This is usually going to be building something. So these are going to be more of your anabolic reactions, while these exergonics are going to be more of your catabolic reactions. You've got a much higher activation energy. So the reactants start on the bottom here, and they have to reach the top before they can come down to the products. We reduce that activation energy, and therefore the net energy investment is less than it would be without the presence of the enzyme. Now, I want you. I went into a lot of detail here, and this is for you to help you understand like what's going on in chemical reactions. I don't actually need you to know everything about this graph. I do want you to like know the difference between exergonic and inorganic, and understand how activation energy is changed with the addition of enzymes. Now you can kind of ask yourself this question: which of these likely represents an anabolic reaction, which represents a catabolic reaction? And so if we get a net gain of energy, which we have here, as our product is going to be down here, we have a positive delta G. This is essentially your gain of energy. We have a negative delta G over here. And so since this is positive and our reactants start with more energy than our products, then we actually gain the energy in between this space. So that's going to be catabolic. And then over here, we're getting a net loss of energy, a negative delta G. That means that when we get up here and down here, we're going to have a loss of energy, probably because we're building something up. That's going to be anabolic. All right, so how do these enzymes do their job? Well, without enzymes, there's this idea of collision theory where two substrates will eventually come into contact and you will get a product, right? But the chance that they collide at the right amount of energy and in the right position is unlikely. And so what an enzyme does is it has an active site, which we see here, and it can fit your substrates in there, whether it's a single substrate being broken down or two substrates being built up. It's going to bring them into a advantageous position to where the chemical action can occur. So here, where we're building this molecule up, we see that our two molecules are close together. It means that it's easier to make our product, and then our enzyme is left alone. Here we've got an example of how the enzyme is going to interact with the substrate. As I mentioned a moment ago, the enzyme has an active site, which we can see kind of here. When it comes in contact with its substrate, you're going to get the enzyme substrate complex, right? So that's what we see here. And while these things do, need to fit together decently well, the enzyme is flexible enough to mold to the substrate. And the idea of it doing that is included in the induced fit model, basically saying that it's going to kind of mold itself around it. Now, if the shape of the substrate is too different from what it's supposed to be, it will not be able to mold itself around it. And so when you get the formation of the enzyme substrate complex, you position the reactants in a way that make it advantageous or more advantageous to perform the reaction. And these enzymes are also going to provide the activation energy required for that chemical reaction to start. And one of the ways they do that is by stabilizing it through that transition state. So when we go back and look at this graph, they stabilize it. This is where it's most unstable. And then when it hits this transition state, it's going to come down and make the product. And so they help position it in a way that it's stable during that transition. Now, enzymes can also require some help sometimes. And so there are things called cofactors and coenzymes. Some enzymes have these cofactors and coenzymes, some do not. These are non-protein components, and oftentimes they're going to be metals and stuff like that. So if not all enzymes have a cofactor or coenzyme, but the ones that do, if they don't have their cofactor, they're called an apoenzyme, which is in an inactive form. And when they do have their cofactor, like we do over here, they're going to be called a holoenzyme. Okay. And generally speaking, you do not get the formation of your product when you don't have a holoenzyme. So apoenzymes don't generally make the product. And so you might think back to one of the enzymes that we talked about in the last chapter, which uses copper as a cofactor. It's involved in aerotolerance, right? And that would be superoxide dismutase. Some versions of it use copper as a cofactor. Now, there are a couple of different ways that these can work, right? So here I've got an image where it kind of changes the shape of the active site in the holoenzyme. You can have a bunch of different things, usually metals like iron, zinc, and copper. You can also have organic cofactors. Again, these are called coenzymes. These are usually things like vitamins. A lot of the B vitamins, in fact, are coenzymes. Here's a list of how different B vitamins are used as coenzymes. Many of them are involved in electron transport, which we'll talk about more in a subsequent lecture. And so we have a bunch of different cofactors that can be used in this electron transport. We've got NAD+, NADP+, flavin mononucleotide or FMN, FAD, coenzyme A. So all these different things are used as electron carriers. And so what we see in this image is that each of these are showing how electrons are being transported, right, through this electron transport chain to generate electrons at the end. Electrons and protons. We'll talk more about that when we get to aerobic and anaerobic respiration. There's a special sort of organic catalyst called the ribozymes. They are made of RNA molecules, but they only have been shown to function on our other RNA molecules. So an example of this would be the rRNA molecules like 16S, 5S, those guys, 23S. They are going to have some catalytic capacity inside of the ribosome during translation. And you can see they have these very complex secondary structures, even though these are still single-stranded RNA molecules. All right, so now enzyme activity can be affected by a bunch of different environmental factors, including temperature. So at low temperature, your cells are not going to grow very fast. Enzyme activity is going to be low. Enzymes aren't going to be able to generate enough kinetic energy to reach the activation energy they need to perform the reaction. As you warm up temperatures, this is going to generally increase your activity up until you get to the optimal temperature. At this temperature, you get optimal activity, highest activity, but you get this very, very steep drop off, right? And that's one of the reasons why we have steep drop offs on those mesophile, psychrophile, thermotolerance graphs, is because enzymes lose their functionality very rapidly above their optimal temperature. And so why is that? One of the reasons is that you get protein denaturation at elevated temperatures. And so here's an example of the active protein in all of its tertiary and quaternary structure. But once we denature it, we start to unravel that structure, getting down to just the protein sequence or the amino acid sequence. very similar things happen with pH. Um, so the concentration of protons and hydroxide ions are going to significantly impact enzyme activity. And therefore, microbial cells will spend a lot of effort in maintaining their internal pH as close to neutral as possible. And so oftentimes the cells don't actually, their pH doesn't, their internal pH doesn't match their external pH. But these enzymes are still going to have like an optimal pH that they operate in. And if you get too basic or too acidic, it will potentially denature that protein, causing a loss of function. And so, in this question, I'm asking like, what would happen between 10 and 50 degrees C? I think this is a good question to ask yourself outside of class, and maybe we'll ask it in class as well, and you'll be prepared to talk about it. But just kind of think about it. If you're a mesophile, what's happening to your enzymes down here at 10 degrees? What's happening to the enzyme reaction rates and the enzyme structure? As you come, we see that growth rate increases along this line. What's happening to the enzyme activity along this line, all the way up to 50? Think about bringing that to class, and let's try to talk about that on Tuesday. Enzyme concentration, unsurprisingly, is going to affect activity, right? So if we have more product than we have enzyme, then we're going to have saturated our enzymes. And this can, result in slower activity because we need to free up the enzyme before we can break down the rest of it. If you have higher unsaturated enzymes, then you can actually perform the reaction faster. The key for the cell is trying to maintain that optimal level of enzyme availability so they're not wasting their growth potential, but they're also not building a bunch of enzymes that aren't being used. So here's another good question to think about for next class is how would you expect enzyme concentration to change between lag and log phase? Maybe even think about how it would change in something like the lac operon. So phosphorus is an incredibly important molecule as it pertains to metabolism. And it can induce these small structural changes on an enzyme. And those small structural changes can affect enzyme activity and function. And it's not a all-encompassing rule. Some are more effective with phosphate attached. Some are more effective without phosphate attached. But you will see a lot of times, you'll see things like kinases and phosphatases. Kinases are going to add phosphate groups through the process of phosphorylation, and phosphatases are going to remove phosphate groups by dephosphorylation. There's something known as competitive inhibition, where you can have a substrate that competes with some sort of inhibitor that can also fit into the enzyme active site. So if the inhibitor is taking up the active site, then the substrate cannot, and therefore it does not, it doesn't get enzymatically changed, right? It doesn't, the products don't get made. However, if there is more substrate than there is inhibitor, then the reaction is likely to proceed without too much trouble. This kind of leads into the idea of allosteric regulation. So you can have allosteric inhibitors or activators. And so what they do is they bind to a site of the enzyme, and that is going to change the function of the enzyme. It's either going to stop function, which would make it an allosteric inhibitor, or it's going to increase function, which would, or turn it on, which would make it an activator. All right. And then last thing I believe I have on this is feedback inhibition. So your enzyme activity can slow down or turn off based on the production of the product. So this should make a little bit of sense if you start thinking about things like the operons that turn on and turn off based on the concentration of a product or of a substrate. So if you, for example, are making tryptophan and you make enough that you can start affecting transcription of the trip operon, then you're going to slow down the production of the enzymes necessary to actually make the tryptophan. So that'd be one way of affecting it from a gene regulation standpoint. Alternatively, when you've got an end product from the production from some sort of enzymatic reaction, it can also act as a inhibitor of the enzyme itself. So you can have some allosteric interaction there. And so this brings us back to our objectives. You should be able to describe the structural and functional features of enzymes and state why cells need them. Describe how enzymes lower activation energy, give examples of coenzymes and their functions, and describe what ribozymes are, how they're different from typical enzymes, and how environmental changes affect enzyme activity. All right. I think it's a good idea to start thinking about these if you're not already doing it. When you're done with these videos, look at these objectives, think about how you feel about them right now, and write down some questions you may have that we can talk about in class next time. With that, I hope you enjoyed, and I'll talk to you all soon.